Good afternoon, good evening. Thank you for the opportunity to present at this webinar. The topic or the title of the presentation is IGHV SHM, Somatic Hypermutation Analysis from Sanger to NGS. So I will briefly share my experience in setting up this assay and how we transfer to next-gen sequencing using in vivo scribes ready-made kits. The disclosures is that this presentation is sponsored by In Vivo Scribe. Now, before we start dealing with somatic hypermutation analysis, I think that we need to explain briefly what is at stake here. What is the target area of DNA that is used for this analysis? The targeted area is the variable region of the heavy chain of the immunoglobulin. All B lymphocytes produce immunoglobulins. And at first, I will present a simplified version, and then we'll just view what is the actual locus look like. But for all intents and purposes, we will present a simplified version. So every cell in the human body has on chromosome 14 the gene for the immunoglobulin heavy chain. The gene is non-functional in its germline form. So in order to produce a functional immunoglobulin, this gene needs to be rearranged. And this is one of the first things that happens in every B lymphocyte that gets a signal to start differentiating in the bone marrow. So on top here, we see the germline version of the immunoglobulin heavy chain. And as you can see, there are various so-called modules. There's the V modules, V for variable. There's the D modules for diversity and J modules for junction. And at the very right-hand corner, we have the constant region of the immunoglobulin chain. If we take, for example, one B lymphocyte in the bone marrow, it's immature. It gets a signal to differentiate to become a mature B lymphocyte. One of the first things that it will do is actually rearrange and cut the chromosome in order to produce a functional immunoglobulin molecule. I should mention that V1, for example, has a different sequence from V2, which is different from V3. Same for the D regions and same for the J modules. So as you can imagine, B lymphocyte randomly will pick one V region, one D region, and one J, and will splice them together with the constant region. So B lymphocyte number X, for example, randomly will choose V1, D1, and J1 and patch them onto the constant region to produce a functional immunoglobulin heavy chain molecule. B lymphocyte number Y at some point also gets a signal to differentiate, and one of the first things that it will do, it will randomly assemble a V, a D, and a J region. In this instance, it's a V3, a D2, and a J2. Now remember, the V regions, the D regions, and J regions differ in sequence somewhat between themselves, so you can well imagine that the VDJ rearrangement here will be of a different sequence and of a different length than the VDJ rearrangement in the other B lymphocyte. The same goes true for a third B lymphocyte, the Z B lymphocyte. It will choose randomly a V2, a D3, and a J3. So yet again, the sequence and length of this region will be different from the others. And this goes on and on for all the B lymphocytes that differentiate. So you can imagine the quantity of permutations and combinations that can result in the vast array of immunoglobulin molecules that recognize different antigens. Now, since the VDJ regions have a unique sequence, you can imagine that this could be sort of a fingerprint, an identity fingerprint of that B lymphocyte. So the unique sequences are unique fingerprints that don't repeat themselves. Furthermore, as the B lymphocyte matures, it will actually mutate or hypermutate some sequences in the V region. And those are actually the somatic hypermutations, which play a role in CLL, the diagnosis that is done by this assay. So the V region, as I mentioned, will contain certain mutations, either insertions, deletions, or inversions, which will still further differentiate the sequence and length of the VDJ area. This is in reality what the whole locus looks like, so a bit more complex. As you can see, there's over 40 different V fragments. There are several D fragments in this area. And at the very end, we have the small, the J region, which is enlarged over here. So as you can see, it's quite complex. And because there's such a variety of V, D, and J fragments, the combinations are enormous. We use the polymerase chain reaction in order to amplify and then study all the various VDJ recombinations present in a sample. For CLL, it is a blood sample that's usually taken, and the first step to do is to extract DNA and then amplify using common primers. We amplify all of the VDJ recombinations in that specimen. So you can imagine that if you have 1 million B lymphocytes, you will have practically 1 million different sequences that you all amplify. As we know, B-cell lymphomas, or CLL, also need somatic mutations in other genes that will drive a certain cell to have growth advantage and give you an overpopulation of that one clone. Just as in any other tumor cells, it's a monoclonal, ideally a monoclonal disease. Therefore, you can imagine that some mutations will give growth advantage to that cell, which will then multiply and expand to give a clonal population, marked in red here, mixed in with normal polyclonal B lymphocytes. And it's exactly the VDJ fingerprint that allows us to follow and study the clone, the predominant clone present in the general population of B lymphocytes. So in a case of CLL, we can imagine that the blood contains normal B lymphocytes all around here, each one having its own specific VDJ recombination, but it will also contain a population of clonal cells which started or expanded from the original cell that became malignant, which has a very specific VDJ recombination, and this combination of VDJ will be transmitted to all the daughter clones. So you can imagine that the clonal population will have a fingerprint signature different from all of the other polyclonal cells. So all have an identical VDJ fragment. How do we go about analyzing this, V region present in the clonal population? Well, we do a PCR analysis or a PCR amplification of all the VDJ recombinations of all the cells that are there. And we can basically obtain a polyclonal smear. So this example is taken from an actual blood sample that does not have a clonal population of B lymphocytes, which means that the smear that we see on polyacrylamide gel is actually made up of bands or amplicons, each amplified of a different VDJ recombination. So there's not a predominant clone there. This is a normal polyclonal population. However, if we look at this example here, we can still see the background smear, the polyclonal population that's present in the blood. However, we do have one predominant band or one predominant VDJ size, and this is actually the VDJ recombination of the clonal population. So now, since we have a band, we have a predominant population, we can directly sequence this fragment in order to determine what is the V sequence of this clonal population. So this fragment can be sequenced. All one needs to do is do Sanger sequencing and determine the sequence of the V region. If we jump back to the disease at hand, which is CLL, one of the predominant markers of prognosis and outcome is whether the V region is so-called unmutated or mutated. We know that the unmutated form of the clonal population has a rather low survival rate of about 9 years, whereas if the V region is considered hypermutated, well, then the survival is a lot longer, 17 to 24 years and more. So what determines whether the V region of the clonal population is unmutated or mutated? Here are the definitions. When something is non-hypermutated or non-mutated, it means that the sequence of the V region of that clonal population in that patient is 98% or more homologous to the actual germline V sequence. If you remember, I said that during the maturation of the B lymphocytes, there are additional mutations that are introduced into the V region in order to expand the actual structure of the various immunoglobulin molecules. Well, this is a hypermutated state. All we need to do is to sequence the V region of that hypermutated state and see if it differs by more than 2%. If it differs by less than 2%, it means that it is 98% homologous to the germline version. Then this is a non-mutated state or unmutated state, and it's a poor prognosis. If the sequence has less than or equal to 98% homology to the germline V sequence, then we call this situation a hypermutated or a mutated state, and this is actually a good prognosis. There are presently two techniques. One is the older, still the gold standard, the Sanger sequencing, and since a few years, next generation sequencing is beginning to take over the analysis method because it has certain attractive characteristics that make it better suited for this sort of analysis. PCR and Sanger sequences is quick and inexpensive, but it could be problematic, as we'll see in a couple of minutes. Next-gen sequencing is, of course, a bit slower. Turnaround time is a bit longer. It's more costly. However, it avoids some of the problems that we encounter with Sanger sequencing. So let's have a look at the actual technique. As you can see here, one VDJ rearrangement on top. The classic way to amplify is to use a primer that's specific for their J region and a primer that is specific for the inside area of the V region. So the amplicon will start, of course, at this primer and will end at this primer. So if we do use Sanger sequencing to read this sequence, we can only read what has been amplified, meaning this amplicon, which inevitably will lead us to miss a certain area of the 5' sequence here. It could be as much as 5 to 10%, depending on where the V primer that is used for amplification is located inside the V segment. Now, if you're trying to compare the sequence of the amplicon to the germline, well, you will not be able to compare these sequences because they are absent. What needs to be used is so-called leader sequences that are sequences where primers bind outside of the V region or on the border of the V region, so that your amplicon can contain as much as possible of the V region, so that you could compare the whole V region to its whole sequence in the germline form. So let's have a look at a few examples that were done by Sanger sequencing. So here we have 7 clonal populations, so 7 CLL cases, with the 8th one being our negative control, regular blood. As you can see, each specimen of blood contains a band, which means a clonal population of B lymphocytes, and these are the fragments that we will be sequencing. Now, if we have a look at sample number 7, it appears to be very homogeneous, a single band, a single sequence, and we can directly sequence this amplicon. And by Sanger sequencing, we get this electropherogram. So we have the sequence. All we need to do is to copy that sequence and pop it into an analysis website. These are all publicly available databases, very rapid. One of them, the foremost, is the IMGT VQuest, which is the best curated database for immunoglobulin and T-cell receptor genes. So all we need to do is to pop in the sequence, press a couple of buttons, and within a few seconds, we will get the result. So here's the sequence that we put in, and the analysis has compared the sequence from our patient to the germline database of all the possible V, D, and J fragments, and it tells us that our specimen, the clonal fragment, it contains the IGHV 333 allele type 1 or 333 allele type 6. The D fragment is the D613 allele 01, and the J fragment is the J402. Now, if we compare the V fragment of our specimen to its germline form, we see that it's 100% homologous. That means it's unmutated. Therefore, this patient has an unmutated V region. Therefore, this patient has a poor prognosis. We can do the same thing on the IGBLAST website from NCBI, and we will obtain a very similar result. And again, it's just formatted in a different way, but nevertheless, you can see that it is IGHV 333, allele 01. You have the D fragment, the J fragment, and of course, over here, you end up with a 100% homology to the germline version. So the results are identical. And for the vast majority of cases, the results are identical. If we look at a sample that does not have a predominant clone, therefore we amplify all the VDJ fragments available, well, of course, there's no predominant fragment to sequence, therefore we get gibberish. I mean, this is obvious, and you can then report that there's not a predominant population of B lymphocytes in this sample. The problem arises with Sanger sequencing is when you have biallelic or biclonal or polyclonal fragments. Most of the time, this results from both alleles on chromosome 14 being rearranged, one giving a non-productive immunoglobulin, then forcing the second allele to be rearranged. And this becomes problematic because if you sequence this specimen, of course you will get gibberish. These are both sequences from both clones being overlaid one on top of another, and you don't get a conclusive sequence. So you can't analyze this one. However, this is where next generation sequencing comes in and actually provides a result where by Sanger sequencing it would be difficult to obtain. The only way that you can make sense out of such a biclonal rearrangement is to actually go back to your gel with a razor blade, slice and separate both fragments, both bands, and elute the DNA from both bands, and then sequence each one individually. This is time consuming, but it can be done if you don't have access to next-gen sequencing. So let me present how these problems are solved by NGS and by the kit that we used. So the software is provided with the kit, so you don't need to go to any external websites. However, I still do because I like to double check my results since these are clinical samples. There's a lot of data that is given, but it's easy to navigate. So for example, when we sequence one clonal population, the first result is that we get the number of fragments that were sequenced. In this instance, over 350,000 V fragments were sequenced. The next piece of information, it gives you the coverage of the V region that was obtained. So 100% of the V region was sequenced. This is obvious because leader sequences lie outside of the V region, so you should be able to sequence the whole of the V region. The actual sequence is given to you of the clone, and now it's truncated over here by double-clicking on the sequence. You actually get the whole sequence. In this case, 492 nucleotide long. Afterwards, the analysis is done and tells you this is the IGHV 439 allele 1 that is represented by this V fragment. You get also the J fragment. It tells you also, well, is the sequence in frame or out of frame. Only in frame productive alleles should be considered for hypermutation analysis. So here it's in frame, that's good. There are no stop codons. So yes, no stop codons, meaning this is a fully productive allele. And now we go to the percentage of this fragment present as opposed to all of the other 350,000 fragments. So you can see over 60% of all of the 350,000 fragment sequence are exactly this clone. So this is a very good illustration that you do have a clonal population of B lymphocytes with precisely this rearrangement. And now this is the business end of the analysis. So in this column, the software compared the sequence of the V region to its germline version present in the database, and it compares by how much does it differ. In this case, there's no difference. It's 100% homologous, meaning it's a poor prognosis for this case of CLL. In graphic form, you also get this, whereby you can see the actual green bar here representing your 60% of the clones that are present. This is an example of a biallelic or biclonal oligoclonal population. So you actually have two predominant populations of lymphocytes, which have either this rearrangement or this rearrangement. Remember, in Sanger sequencing, these would be presented as two closely similar bands on a gel. By next-gen sequencing, you actually separate these results out. There have been several papers that were published that guide a person to how to analyze these sequences. And for example, there's one from 2011. There's an updated recommendation in 2016, and the most recent one, which has the updated ERIC recommendations, ERIC being the European Research Initiative on CLL, the body, the specialized body that carries out a lot of research in this field. So they have recommendations how to analyze and how to report these IGHV sequences. So let's look at one case where we have a major clone and a minor clone. As you can see, both are almost identical in numbers, 34%. So most probably we're talking about a biallelic rearrangement of a single B lymphocyte clone. How do we analyze this? Well, here we go. So the major clone, we can analyze the sequence, and we see that it has no stop codon. It's in frame, therefore it's productive. It has an open reading frame. It's the IGHV 169 allele 01, and it's at 100% homologous, so it differs by 0%. However, we have a second allele here, which is different. It's the IGHV 333 allele 1. It's 100% homologous also, but we notice that it has a stop codon. Therefore, according to the guidelines, only productive alleles are used to analyze and report hypermutation analysis. Therefore, we ignore the second allele. We only take into consideration the productive allele. Therefore, we report this as non-mutated. In this case, both alleles are the same, but sometimes they differ. So you only report the productive allele. So here, there's no dilemma. The major clone is productive. There's no stop codon. The minor clone is non-productive. So it's not considered for SHM analysis, and we report this as non-mutated or unmutated. Here's another case in hand where we have a slight difference in the proportions of one clone as opposed to the other, 41% to 28%. And again, we do the analysis, or the software does the analysis. We take the major clone, which is the IGHV 434 allele 1. It is, I will talk about this in a couple of minutes. It is a stereotype subset 4. Which is, by definition, an indolent allele. It's mutated, so that's a good prognostic marker, but it has a stop codon and it's not in frame. So we should not consider this. It's mutated and indolent, so it's a good prognostic factor, but we do not consider that clone. We go to the next clone that we see here. So it's a minor clone, 28%. There's no stop codon. It's in frame, so it's a productive allele, IGHV 37201, and it is unmutated. It's above 98%, so it's unmutated. So therefore, poor prognosis. So here, we would perhaps see that there's a conflict between the two alleles. However, since the initial one is non-productive, we don't consider that. So we report this as unmutated, therefore a worse prognosis. It was noticed by the researchers at ERIC that some alleles, whether they are mutated or unmutated, they have either a good prognosis or a poor prognosis, regardless of the mutation status. And this is another website that is used to analyze the stereotype subsets. So all one needs to do is to copy the sequence into this website, and within a few minutes, one will get the result, whether it belongs to one of the stereotype subsets. There are basically 5 major ones. Subset number 1 is poor prognosis and has an aggressive clinical course regardless of its mutation status. Same for subset number 2. Subset number 8 also has a higher risk of Richter's transformation. And the major subset number 4, there's also number 5, which have an indolent course regardless of the mutation status. Now, there's a lot of papers that are being written presently to explain what these subsets are. In short, these are the shapes of the actual immunoglobulin molecule, the variable regions that interact with antigens which seem to stimulate the growth and progression of CLL. So there's a lot of antigen stimulation that is happening, and depending on the sequence and the shape of the immunoglobulin molecule, not necessarily on its mutation status, that will predict either a good or a poor prognosis. In order to navigate in all of this, there's one good paper that was published in Leukemia in 2017, which basically gives you this tree, how to follow the results that you get. I had one of my residents work on this flow chart, this flow chart, how to analyze results. So by following the line, it basically tells you what needs to be done and how to report a result. So I will end on this point. I thank you for watching this video, and if you have any questions, I'm sure that there will be some people that we will be able to answer any of your questions. Thank you very much. Have a good day.
Well, hi everyone. I'm glad to be with you, although I wish we could be doing this in person. I'm Ryan Schmidt. I'm a molecular pathologist at the Children's Hospital Los Angeles, and I'm excited to share with you some validation work of a minimal residual disease assay for lymphoblastic leukemia that uses in vivo scribe lymphotrack kits. Before we begin, I'd like to disclose that I've served on an advisory board for Adaptive Biotechnologies, but I don't have any disclosures relevant to in vivo scribe, except to say that they've been a great vendor to us and I appreciate what they do. I'll begin with a little background about Children's Hospital Los Angeles. So we're a large pediatric center for lymphoblastic leukemia diagnosis and treatment. We perform approximately 1,000 flow cytometry-based MRD PET tests per year. All patients are currently assessed for MRD by flow at multiple time points. We are a COG-approved regional reference lab for patients enrolled in COG clinical trials. So given the needs of our institution, we wanted to stay ahead of the curve for MRD testing and thus considered developing an NGS-based MRD assay. We noticed the potential for increased sensitivity over flow, as well as the potential for increased prognostic significance of a negative result. After evaluating several kits, we chose in vivo scribe lymphotrack. In vivo scribe has long been a leader in molecular clonality testing, and we were drawn to the DNA-based analysis given that gene expression is not required for detection of the rearrangement. And removing expression as a variable also makes the interpretation a bit more straightforward. The end result becomes the percentage of total reads, and that can be converted to number of cells using the internal control lymphoquant spike in. Additionally, DNA gives you the potential for better sample stability, and you're potentially more likely to get a result from archival specimens. So we designed a strategy around detecting IGH and TRG gene rearrangements since previous reports in the literature and from colleagues told us that a large proportion of lymphoblastic leukemias have these genes rearranged. For IGH, we chose to use the Framework 3 kit since we were focused on obtaining the sequences of the junctions and the leukemic cells had not undergone somatic hypermutation. So the strategy has two parts to it. There is the first part of determining the clonality and trackable sequences at the time of diagnosis. So what we do for that is we sequence the specimen with the IGH Framework 3 and the TRG kits. For specimen types, we accept bone marrow aspirate, peripheral blood, and FFPE clot sections. We aspire to test aspirate slides, but we have not yet done that. In terms of sequencing depth, you only need a low sequencing depth at the time of diagnosis, and for DNA input, we use 200 nanograms, although we've shown that you can use much less if needed. Contrast that to the MRD monitoring step. So for that one, we're going to test using the gene corresponding, gene or genes corresponding to the sequences that were identified at the time of diagnosis. So again, we're trying to do minimal residual disease monitoring for a known rearrangement sequence. For specimen types, we use bone marrow aspirate as well as peripheral blood. We're going to sequence very deep and hope to achieve a sensitivity that's better than 1 in 10 to the 5th cells. And to do that, we need a lot of DNA input on the order of a few micrograms. So our laboratory has several of the Illumina sequencers, and we chose the IGH Framework 3 and TRG kits partly because their amplicons are compatible with the 300 cycle kit. So that allows us to use the NextSeq, and it allows us to use the several different scales of the kits for the MySeq. So you can see listed there, nano, micro, and the standard MySeq V2 kit. With the nano kit, if you're just doing the test at diagnosis, you're able to run 48 libraries and get approximately 40,000 reads per library, and that's sufficient. The kits come with up to 24 indices on Illumina, and you can use the same index and sequence for both libraries of both genes on the same run. That's what this schematic is trying to show you here. So that allows you to do 24 libraries for both IGH and TRG. We're illustrating here 22 plus a positive and negative control. So up to 48 libraries together on the same Illumina run. And also, I didn't mention this before, but having that shorter amplicon potentially gives you better FFPE compatibility if that's a specimen type that's of interest for you. So a quick note about sequencing for MRD. We want to make sure that we obtain enough sequencing reads and use enough input DNA such that we obtain a very high sensitivity and avoid false negatives due to sampling error. So here's some modeling that's been done by InVivoScribe. It helps you think about this. And we want to operate on this green line here, slightly to the right of it, and on that green curve towards the flat part up here. So we want to obtain, in doing an MRD test, on the order of a few million sequencing reads. So for each microgram we sequence, we try to obtain 1 million sequencing reads. And we'll go into more detail on that in subsequent slides. So we had to define kind of what a positive result would be in the diagnostic assay. And for lack of a better term, we're calling this a clonal sequence. So this is defined as a rearrangement sequence that's present at ALL diagnosis that represents greater than 5% of the total reads for either the IGH or TRG genes. The idea of this is to identify sequences associated with the dominant leukemic clones that are suitable for monitoring by the MRD test. And you know, it's an arbitrary cutoff, but there is some rationale behind it. It's at a level that's consistently above the normal polyclonal background. And diagnostic samples with lymphoblastic leukemia have over 20% blast and typically over 50% blast in our hands. So thus, the sequences that represent over 5% of the total reads, they're likely to be associated with the leukemic clones and the if it's an oligoclonal process, the major clones. So this allows us to focus on the sequences that are most relevant for monitoring for disease. So one of the first things we set out to do was to determine clinical sensitivity. And what I mean by that is to figure out the proportion of our patients with lymphoblastic leukemia where we could identify these clonal sequences in either the IGH or the TRG gene. So we took 69 diagnostic B-ALL DNA specimens that represent all the diverse subtypes, cytogenetic and molecular subtypes listed there. And when we did this, we found that 97% of those specimens had at least one TRG or IGH clonal sequence. And in the graph that you see below, it's a little busy, but it shows you that occasionally we would see oligoclonality as well, where you would have more than two clonal sequences identified for one of these two genes. That wasn't totally unexpected. We also sought to determine the clinical specificity. So would we identify sequences above the 5% threshold in unaffected samples? So we had 5 unique unaffected peripheral blood samples, and testing them identified no clonal sequences above that threshold. We had 24 ALL negative bone marrow aspirates. These are from individuals with a variety of conditions that had morphologically normal bone marrow evaluations, including individuals with ALL that were in remission. And from those 24 specimens, we only had one sequence that crossed the 5% threshold as an IGH gene in a specimen from an individual with a history of Hodgkin lymphoma that had pancytopenia and a hypocellular bone marrow. So perhaps it's not surprising that there was this sequence present above the 5% threshold, given that their bone marrow isn't completely normal. For the MRD test, we did some input DNA titration. Of course, the goal is to test as much input DNA as possible, so as many cell equivalents of DNA as possible. The kit procedure specifies that you could use up to 2 micrograms of input DNA per library prep reaction. Ultimately, we chose 1 microgram of input DNA because what happens is once you reach a certain level, it's going to interfere with your amplification. So for us, we were able to get good amplification with 1 microgram. So for each reaction that we perform, we put in 1 microgram of input DNA. If we want to test more than that, we would line up multiple reactions in parallel. So for MRD, we wanted to maximize the input DNA that we could put in for the greatest possible sensitivity, but recognize our practical limitations. So cost, sequencing capacity, number of library prep reactions, and the available input DNA. And in doing this, we sought to achieve at least 1 in 10 to the minus 5th sensitivity. And to do that, we assess approximately 600,000 cell equivalents. That's 4 micrograms of input DNA. And we attempt to sequence at least 4 million reads from that. So we performed some analyses for precision, so reproducibility and repeatability. We did samples for both the IGH and TRG genes for intraday, interday, interoperator, and inter-instrument reproducibility. Each of these were run 3 times. And in doing that, our results were 100% concordant, and the coefficients of variation for the quantitative estimated percent MRD estimate were relatively low, less than 25% for all of them. So it gave us a reproducible measures. We did serial dilution studies to look at limit of detection as well as linearity. And the way we did that was we took diagnostic specimens and we diluted them into pooled peripheral blood. And then ultimately, we measure the estimated percent MRD, as I alluded to on the last slide. And the way you do that is you first calculate the estimated cell equivalents based on the lymphoquant internal control spike-in, which is equivalent to 100 cell equivalents of DNA with a rearrangement sequence that you spike in. And then you take the fraction of the estimated cell equivalents over the total cell equivalents. And there I see a small typo here. This should be times 100% to give you the estimated percent MRD. All right, so when we did this serial dilution of a specimen with a IGH rearrangement, we show the following columns up here. So the number of reads that are an exact match to that that clonal sequence identified at diagnosis, the total number of reads obtained, the number of reads obtained for the lymphoquant internal control, the calculated estimated cell equivalents, and the percent MRD. The first thing you notice here is that this number might be a little high than what you might expect for a 1:100 dilution, but that happens when you dilute into a different specimen type. Here we have the leukemic bone marrow aspirate diluted into the peripheral blood. So sometimes you see a jump at that first dilution point, but from there on, it was highly linear and quite sensitive for the linearity. The R-squared value was greater than 0.99. You're seeing here on a log plot. Same kind of results for the TRG clone. The R-squared was even higher this time. Interestingly, in this one, we ran a 1 to 10 million dilution, and we saw a few reads with the exact match. I think it just shows you that you can get a low level of these reads due to the index hopping, so kind of bioinformatic contamination, as well as potentially low level introduction of the sequence in our pipetting steps. So the message there is just that interpret with caution when you find a very, very low number of these sequences. It's kind of similar to what we do in our flow lab. There's a level at which the percent MRD is reported out, and below that it's kind of a indeterminate presence of a low number of events. But the end conclusion is we reached our goal that it was sensitive down to 1 in 100,000 and highly linear. So we did a comparison with specimens that were tested in our clinical flow lab for MRD, and we had 38 total specimens. There were 19 flow MRD positive specimens, 19 flow MRD negatives, where they had clonal sequences, a clonal sequence or clonal sequences identified in the associated diagnostic sample. And doing that, we assembled the 2x2 table that you see at the bottom there. We found that the test was highly sensitive, so sensitivity of 94.7%. We had one discordant specimen that was positive by flow and was negative by the NGS MRD assay. Perhaps that is due to there being minimal residual disease from a clone that lacked a rearrangement. I'm not quite sure, but it does point to the potential importance of multimodality testing. And then for the specificity, it was 73.7%, but I put an asterisk here for these 5 so-called false positive samples where there was an NGS MRD positive result, but the flow MRD was negative. And we'll come back to that in a second, but we have to keep in mind that the NGS test might be more sensitive than the flow gold standard. So in comparing the percent MRD estimate between flow and NGS for those specimens that were positive by flow, we actually found that the numerical estimate, it was quite concordant. So you can see, you could almost draw a line through all of these points right here. So in conclusion, there is good quantitative agreement for the percent MRD estimate. So now coming back to those 5 potentially false positive NGS MRD samples. So, you know, it's been reported that NGS might be more sensitive than flow cytometry based MRD. And we noted that these 5 had a percent MRD estimate that was less than 0.01%, which is below our stated flow cytometry sensitivity. So this is consistent with the possibility that we're seeing true but very low level residual disease that's below the sensitivity of flow. But the significance of that, at least at this time, isn't totally clear. There hasn't been extensive follow-up on these patients, but none of them have relapsed yet at this time or become positive by flow MRD. So we'll have to wait and see. So to summarize what I told you today, the in vivo scribed lymphotrack, IGH framework 3 and TRG kits allow for lymphoblastic leukemia MRD monitoring. We found them to be easy to use and flexible. We had a high clinical sensitivity for detecting clonal sequences in our B-ALL patient population, and our MRD analysis, it was reproducible, it was linear, and it was potentially more sensitive than flow cytometry based MRD. So with that, I will wrap up, and I want to acknowledge all of my colleagues that I listed here that did a great deal of work behind the scenes to give you all the results that you saw today. So thank you so much for your attention, and I'll stop there.